we are going to start with the second day of this AI conference, which is sponsored by Sir. We have with us Dr. M.A. Budkar Sir, who is the chairperson for this session. Dr. Budkar Sir has completed his graduation and post-graduation. So he is working as a postgraduate teacher as well as a PhD supervisor at Shivaji University, Kolhapur. He has guided more than 45 students of PG. Sir has received the Best Research Paper Award in 2018. He is also a nominee from the CPCSE in New Delhi. He also received of the Young Achiever Award in 2020 from the Institute of the Scholars. Sir has 2 patents granted and 2 published patents to his credit. He has also published more than 70 research articles. Research work citation is more than 850. He has authorized 4 books and he is a co-author published book chapters at the Taylor and Francis as well as the Springer. Sir has also received more than...
It's a privilege to work as a chairperson for the session of Dr. Dadasaheb Patil, one of the renowned persons in the field of research. Presently he is working as Executive Officer, Quality Control Department at Serum Institute. He has completed his Diploma in Pharmacy from Government College of Pharmacy, Karad. His Bachelor of Pharmacy from Government College of Pharmacy, Karad. He qualified in GATE exam conducted by IIT Chennai in the year 2003. Elected as Junior Research Fellow and Senior Research Fellow from DST, New Delhi. Thereafter he was awarded as Senior Research Fellowship from Serum Institute of India Research Foundation, Pune. He completed his PhD from Serum Institute of India Research Foundation and Pune University. After submission of his PhD thesis, he was associated with Symbiosis International University, Pune, where he conducted lab training for MSc as well as PhD students, and he conducted lectures for them on advanced analytical techniques. After PhD, he continued as PDF and worked on plant metabolomics and development of rat and human cytochrome B450 based assays for early prediction of drug-drug and drug-herb interactions. He was associated as a senior research associate in the Institute of Transdisciplinary Health Science and Technology, Bangalore, and work on the projects of biocompatibility and bioavailability of Ayurvedic metallic preparations at Center for Pharmaceutics, Cognosy and Ecology. Then he performed his doctoral work on herb drug interaction studies on Tinospora cordifolia, and he was the recipient of Best Poster Award at 12th International Congress of Ethnopharmacology held at Kolkata. Then he had the opportunity to join the hands-on training workshop on bioanalytical techniques in pharmacokinetic studies at All India Institute of Medical Sciences, New Delhi. He has several research publications to his credit. He has authored some books, patents and book chapters. He is associated with some of the renowned institutions like Lupin Research Park, Pune, CRO, that is CNEPS Private Limited, Pune, Bioanalytical Technologies, Pune, Analytical Solutions, Mumbai. He has well expertise in writing reports, scientific publications and preparing and drafting proposals for grants and lab management activities. He is working as an editorial board member and reviewer for several reputed international journals. He is associated with Serum Institute of India Private Limited from year 2014 till date. And how many of you have received the Covishield vaccine? Just raise your hands. Both the doses. Okay. I will explain you in what context I asked you the question. He has worked as analytical method development and quality control team member, team leader for COVID vaccine. So big round of applause for him, as well as malaria and several other vaccines. So we are very much fortunate to have such a eminent person as a resource person who will be guiding us today. So on behalf of Ashok Rao Mane College of Pharmacy, on behalf of principal, organizers and all team members, once again I welcome you, sir, on this occasion, and I am thankful for giving me an opportunity as a chairperson. Thank you.
To talk on artificial intelligence. So, but what I am going to talk is basically on what are studies which are needed to design and develop artificial intelligence. So those studies are very important. So any design of any software or any database, you need some inputs. And those inputs are very important. So inputs in the sense, your earlier studies, which may be in silico studies, which may be your in vitro studies, studies based on the cells, studies based on the enzymes, those are very important. Those studies gives you some clues, some idea on your research. And based on those studies, you can design your in vivo studies. So those are in the animals specifically. So those studies you can plan and you can derive some, based on your some hypothesis, you design some studies based on your earlier observations from in vitro studies, and you do some studies in vivo based on any animal model like mice, rat, guinea pig, and from that you will generate some concrete results. So you will say, yes, I tested this drug and it is working good for the anemia, it is working good for cardiovascular related complication. But that is not enough. So you need to do it in clinical setup. So you need to do some studies in clinical setup. So that is what we call as clinical trials. So these all data starting from in vitro, in vivo, clinical studies, also the studies from some post-marketing studies. If it is gathered in a manner so that we can take some decision, and those studies are we consider input studies to develop and design some logical database or what we call as artificial intelligence. So I am going to talk to you the very important topic that is integrative medicines. So we will discuss what does mean by integrative medicines, and that I am going to talk to you about important aspect, that is the herb-drug interaction. So when we see integrative medicines, we need to study herb-drug interaction. That is a prerequisite to study and design integrative medicines. And based on these studies, we can go to design of our AI-based tool, so that once you click some of AI tools, you will get the whole database. So if you click one herb, that is from Ayurveda or your traditional medicines. So if you put that name in that database, for example, ashwagandha, so you will get the list of all the drugs. Drug means what I mean is to allopathic drugs. So how this ashwagandha is going to interact with your allopathic drugs. Whether that interaction is useful, whether that interaction is unwanted, whether that interaction will give you some kind of adverse event. So that information, by a click of your system, you will get those data in front of you, in front of as you are a pharmacist, and also you can recommend some of the therapies to the doctor. Yes, you can take one ashwagandha along with some anti-cancer medicines. So those can be beneficial to the cancer patient. So that is why these input studies are important to design artificial intelligence. So before we start actually on the topic, I would like to give a background about my company, Serum Institute of India Private Limited, and it is founded in way back 1966 by Dr. Cyrus Poonawalla, chairman of our company, with a vision that it can supply high quality vaccines with affordable price to all human beings, to all the nations. And yes, of course, the company is progressing well under the leadership of our CEO, Dr. Adar Poonawalla. And we all know that because of the COVID pandemic, we need to manufacture larger number of COVID vaccines. And we have manufactured around 130 crore COVID vaccine in last year, and which have been supplied to India and to other part of the countries. So, by all means, we are the largest vaccine manufacturer across the world. If you can go by the numbers, we are manufacturing about 65% vaccines. When you consider all vaccines, we are manufacturing 65% of vaccine manufactured by the Serum Institute. That in the sense, if you vaccinate the 3 child in the world, out of 3 child, 2 child will get vaccinated from the Serum Institute of India. So that is the coverage and the capacity Serum has. And of course, we mentioned our vision is to supply quality and affordable vaccines, and quality will be monitored by certain regulatory agencies like WHO, and we are respected by several agencies like USFDA, like TGA Australia, and other so many European agencies, MHRA. So we have been audited and respected by those agencies. So this is just the background of our institute. So now we can start about our topic of the discussion. So there is shift in thought. Earlier it was basically we need to treat the disease, and now it has been shifted, we need the health. So there is a paradigm shift of disease to health. So in this, earlier and still we are discovering new drugs to treat the disease, and those new drugs are coming out of your targets, either single targets or multi-targets. So these drugs, definitely we need these drugs, but these drugs are mainly we need in acute conditions. But what about our chronic diseases? So take an example of chronic diseases like cancer, like diabetes. We have some drugs for diabetes and cancer. We have drugs which are responsible for the symptomatic treatment for the diabetes. For example, in a diabetes, it is a metabolic syndrome. We consider it as a metabolic syndrome with hyperglycemia. We have insulin resistance. We have hyperlipidemia, and we have diabetic complication. So we are treating them as a symptom. We have hyperglycemia, we are using some anti-hyperglycemic agent. We have hyperlipidemia, we are treating it as a statins to lower the lipid levels. But we are not preventing the chronic diseases. We are just treating them. So for example, if you have a headache or body ache, you are going to take some painkillers. So what does they do? They just block some receptor in the central nervous system, and you will feel that there is no pain, and you will feel better. But that does not mean you are eliminating the root cause of what is the basis to create headache. So like that, we need a drugs importantly, but we need something bigger, more than that, to treat and to prevent such chronic diseases like diabetes, cancer, you name it, cardiovascular diseases. And that is why the newer concept is coming up, is integrative medicines. So we need some kind of integrations among our existing practices, among the existing therapies. So what is that integrative medicines is, we know we have allopathic medicines, but those are not sufficient. So we need the drugs from other systems. We need the drugs from our traditional medicine systems, like from Ayurveda, like Chinese herbal medicines. We need some herbal medicines. So we need some of the practices to actually regain our health. And those practices, like we need some yoga, we need some meditation, we need a diet, we need a nutrient. So all of this component compromises of the basis for the integrative medicines. And that is what we need nowadays. Okay? So this, you can see, integrative medicines is an evolving healthcare practice which is based on the holistic approach. And as I mentioned, it involves the nutritional medicines. We need to monitor our lifestyle, our behavior. We need to monitor our mind and body medicines, some energy medicines, some of the manipulative therapies, of course the surgeries, and of course we need drugs, pharmaceutical drugs, and along with the other medicines like herbal medicines. So in year 2017, WHO renamed the traditional and complementary medicine as a traditional, complementary and integrative medicines. And the main goal of this integrative medicine is to achieve a state of physical, mental and social well-being. That is the concept of health. And that is how the integrative medicine evolves as a rationale for modern medicine plus herbal medicines. And if you can see in my next slide, so those who wanted to do some research, those who wanted to pursue their career, they have good opportunities in integrative medicines. So you can see the list of universities. These are the around 20 universities from the US. So those are actually working on the integrative medicine, and they are actually practicing the knowledge of integrative medicines for the chronic diseases like cancer, like diabetes. So in cancer, they are trying to use certain herbal medicines, certain kind of the practices like yoga, meditations, so that they can use, of course, the chemotherapeutic drugs or radiation therapies, but they can eliminate the side effect associated with the chemotherapy. So those number of, you can name it, the Harvard Medical University, you have Michigan University, of course Minnesota, Petersburg. So several universities, they have several hospitals at their institute, and they are practicing integrative medicines. So those who wanted to pursue or go with your research career as an integrative medicine scientist, so those can look into these universities. Not only universities from the US, but universities across the China also started integrative medicine practices under their certain hospitals, and in India as well. We have started our health minister recently has an announcement that they are going to start integrative medicine practices in all of our government hospitals across all our India. So again, it is big opportunities for all of us, because we are a pharmacist and we know better than, I think, we know better the drugs and medicines than the doctors. So yes, so for integrative medicines, we need to study the heart-drug interactions very important, because one of the aspects, I am not going to talk about integrative medicines, and there are several aspects which involves the other therapies like practices, but I am going to focus on the important aspect is the heart-drug interaction studies. So, and we know that, so there is concurrent use of herb along with drug is increasingly, we can see there. And one of the studies from the US, they have suggested that 31% patient use the herbs concurrently with the modern medicines or allopathic medicines, and out of them, 70% of them, they do not report whether they have taken some herbal medicines along with the allopathic medicines. So what it leads, there are possible incidences of herbal interactions, and those incidences may lead to the some beneficial or some adverse event. Sometimes those even can be fatal. So that is why very important to study the herbal interaction, the one of the aspect of integrative medicines. So based on that, you can predict, you can actually recommend certain medicines, certain allopathic drugs with certain Ayurvedic medicine or certain herbal medicines in a chronic disease conditions. So when we talk about the interactions, so normally we know that there are two types of interactions. When you take allopathic medicines with herbal medicines, it will cause either pharmacodynamic interactions or it will lead to pharmacokinetic interactions. So when drug interacts with the receptor, it will give certain kind of effect. When drug is given with the herbal medicine, it has a multiple component, and those components can either react with that receptor or some other receptors. It will lead to the synergistic effect, maybe it will lead to the additive effects, sometimes it may lead to antagonistic effect, or sometimes you may not see any effects, so no effect. So that is what we call as a pharmacodynamic interactions. So in pharmacokinetic interactions, we all know when you take a drug, it will absorb to your stomach. Then if you take drug along with the herbal medicines, they will impact your absorption with some kind of enzymes. Also, it may have some impact on the distribution of your drug. So it may have some impact on the protein binding of your drug. So, and in some cases, we know that drug is get metabolized in the liver, but there are certain components of the herbal medicines which can either enhance or inhibit the lipid enzymes, phase 1 or phase 2 enzyme systems. So that may also alter your pharmacokinetics of your allopathic drugs, which when you take up with the herbal medicines, and also of course the excretion of your drugs, because excretion is also mediated through some kind of transporter systems. So like some transporter proteins which are taking out your blood from the blood, and it is clearing it from the body to urine. And if you take some medicines, herbal medicines, they may impact on the excretion of your allopathic drugs. So overall, it may lead, I think it's not clear here. So you can see the plasma concentration profile of your drug. If medicines have some impact, it may increase your plasma concentration profile to certain level, which we call as a minimum tolerable concentration. If that concentration goes beyond minimum effective concentration or minimum tolerable concentration, you can see some kind of side effect, because your drug concentration is high in your blood. But sometimes you herbal medicines have some impact, so that your pharmacokinetic profile goes down. It may be the concentration goes to the minimum effective concentration that is required for the effect. So it will go to the sub therapeutical concentration, and you may not get the desired effect of your drugs. So these are possibilities of your pharmacodynamic interaction. So this is basically about the pharmacodynamic interaction when you take a drug with herbs, how it modulates the different receptor, and also it may have some impact on the pharmacokinetics parameters, because the herbal drugs may have some impact on your enzymes, for example liver. So, and that is why your plasma concentration may alter and you may not get the enzyme effect because of the allopathic drugs. So as I mentioned, what are the targets which we need to look at when we need to study the pharmacokinetic interactions? So there are important targets which we consider as the metabolizing enzymes from the liver. So we know that almost all our drug is get metabolized by our liver enzyme systems, what we call as a cytochrome P450 system, CYPs. So and that also we have selected cytochrome P450 enzyme systems which are majorly responsible for the metabolism of most of the drugs. So in that you can see CYP3A4, which is the largest enzyme responsible for the metabolism. Then you have CYP2D6 and then CYP A2 and the CYP2C19. So these enzyme systems we consider as the major targets because they are responsible for the metabolism of around 50% of your drugs. So when a patient takes the cancer drugs, so cancer drugs are mostly likely to be metabolized because of the CYP3A4 metabolizing system. And if patient is going to take some medicines and if those medicines, maybe those herbal medicines have some impact, either they have impact on inhibition or the enhancement of those enzymes, you may certainly have some kind of pharmacokinetic interaction with the anti-cancer drugs. And also there is a possibility of induction of some enzymes, and that induction happens to the some kind of receptor which is called as a nuclear receptor, pregnant X receptor, PXR, and constitutive androgen receptor, CAR receptors. Those are also responsible for the induction of your enzyme systems. So these are the targets basically. So to study the pharmacokinetic interaction. Also, as I mentioned, there are certain proteins which we call as a efflux protein or efflux protein. So those are responsible for the excretion or the taking out of blood from your cell. Those called as a efflux proteins. So those are we can categorize them as a P-glycoprotein, breast cancer resistant protein, BCRP, bile salt, export proteins, and there are other types of protein receptors which are responsible for influx of your blood inside the cell, and those are nothing but the organic amine transporting peptides, organic cation transporter proteins, organic amine transporter proteins. So these are basically transport proteins involved in your drug modulation. So these can be the targets to study your pharmacokinetic interaction. So what we have done is we know that based on this study we know that the CYP is the major enzyme, hepatic enzyme responsible for the metabolism of most of the drugs. So we wanted to study by using this enzyme, and we are taking the commonly used herb medicines in India. So what is the effect of this commonly used herb on this enzyme system, whether they are inhibiting this enzyme or whether they are enhancing this enzyme system. So in this we have used the Shatavari asparagus racemosus, which is commonly used in India. We have used the Vigania somnifera, which is called as ashwagandha, and then Tinospora cordycelia, which is a guruchi. So these are basically rasayana drugs which have been used along with the allopathic medicines. So we wanted to study whether these drugs have some impact on these enzyme systems. So what we have done is we have taken these enzymes, which is basically human CYP pure enzymes, CYP3A4 in the microplate, and we add this extract and we add the testosterone, which is the substrate, which is because of the CYP3A4, it gets metabolized into 6 beta hydroxy testosterone, and if that level is higher, then we can say there is the metabolism of this testosterone. And if your heart has some inhibitory potential, then there is less formation of 6-hydroxy testosterone, and you would see there is some impact on inhibition of these CYPs. So very important, we need to monitor these levels, as I mentioned, the testosterone as well as 6-beta testosterone, because it is ultimately going to tell you is the potential of your drugs on the enzyme system. So here you can see, is it visible or it's quite hazy? So, and this you can clearly see this is the monitoring peak of 6-beta hydroxytestosterone, and this is the major peak which is testosterone. So your method should detect these two peaks, because you are going to monitor the activity based on this profile. And you should we have tried some pure component in the experiment, that is some of the component isolated from the tinospora, that is alcohol component, which is we consider as berberine and the jepurizine. So those components which are inherently there in your extract or the part of your extract, then those components should not interact with your peak of interest. So there should not be any interferences. So otherwise you would lead to some non-conclusive results. So it is clear that your profile is good. You are seeing the 6-beta hydroxytestosterone and you are seeing the testosterone. So, and then we have incubated all this extract in the enzymes, and we monitor the peak areas of 6-beta testosterone, and we profile the IC50 values based on the percent inhibition versus the concentration of each extract we have chosen for the studies, like Vidalia somnifera, the tinospora, and Shatavari, and also we have used the pure component to see what is the effect of this component. So in that, what we observed is we could see the inhibitory potential of some of the isolated components, that is the berberine, jepurizine, but we do not see any impact of this extract like Vidalia, tinospora, and asparagus on inhibitory potential of CYPs. So based on this, we can say these herbal drugs may not have some impact on the pharmacokinetics of those drugs which get metabolized by CYP3A4. But this is not enough. So this is just our in vitro findings based on the enzyme studies. So we need to prove when we inject this extract to the animals along with one of the model drug which is get metabolized by CYP3A4, that is the cyclophosphamide, and we actually wanted to see whether there is impact of actual interaction when it is administered in the animals, and we wanted to see the pharmacokinetics profile of that drug. So that experiment we have planned in the mice. So we have planned the study with respect to one drug, that is Guruchi tinospora cordifolia, in a dose range of 100 microgram per kg to 1600 microgram per kg, and we have given this Guruchi with different doses by oral route up to 21 days to the mice, and at 21 days we have injected cyclophosphamide, which is an anti-cancer drug, which is, we know it is getting metabolized by CYP3A4, and on the same day after injecting cyclophosphamide, we have collected the blood for different time interval up to 3 hours, 180 minutes, to see what is the plasma concentration profile of your cyclophosphamide at 0, at 10 minutes, at 30 minutes, at 60 minutes, likewise up to 180 minutes. And after 4 days, we have some set of animals. After administration of cyclophosphamide, we have taken the samples for hematology to see whether these drugs, the cyclophosphamide and glucosamine, have some impact on the pharmacodynamic parameters, like as we know that cyclophosphamide is anti-cancer drug which cause the myelosuppressive effect. Because in myelosuppressive effect, your all blood counts like WBC, platelet count goes down, and that leads to the leukopenia or the leukocytopenia. So we wanted to study whether is there some improvement in your blood cell count after administration of glucosamine. So that can tell you whether it has some beneficial effect or it has some adverse effect, whether they are still lowering your blood count. So the study we plan and we monitor, as we mentioned, we have collected blood from different time interval to monitor the cyclophosphamide, and we measured in the HPLC method. We got good fit at cyclophosphamide and also the peak at serum cell rejection time for the cyclophosphamide from the samples which we collected at different time intervals. So based on this, I may not go into details about the methodology. Every time you need to have very good method. Method should be validated as per the international regulatory guidelines like USFDA, like ICH guideline. You should determine the method specificity. You need to know the repeatability. You need to understand the accuracy of your method. These are all aspects of your analytical method validation. Those also taken care while using this method. And next, you can see the plasma concentration profile of your all subjects. So the subject or the animals which are given cyclophosphamide alone and the cyclophosphamide with the kilospora at 100 microgram, at 200 microgram, at 400 mg per kg, mg per kg at 800. So all this profile looks overlapping. So that clearly shows there is no impact of your extract when it is administered with the cyclophosphamide. There is no change in the pharmacokinetics profile. So this tells us that there is no pharmacokinetic interaction of glucosamine with your anti-cancer drug, which you have seen from your in vitro studies, where you can't see any impact or inhibiting potential of your glucosamine on cytochrome P450, that is CYP3A4 enzyme system, and that 3YP3A4 is responsible for the metabolism of the cyclophosphamide. So in vivo studies clearly says that yes, your in vitro finding is in line with your in vivo finding. So there is no interaction. So it is said to take glucosamine along with the cyclophosphamide or anti-cancer drug. So as I mentioned in my animal model, that we have taken some hematology samples to see what is the effect of this glucosamine along with the cyclophosphamide on the leukocyte count or white blood cell count. So you can see clearly. So these are the blue ones which are because of your glucosamine. Glucosamine enhance your leukocyte count, and it is bringing down your normal leukocyte count. And when you administer cyclophosphamide alone, it is decreasing your leukocyte count, and that is why you call it as a myelosuppressive effect. And because of glucosamine, you are reversing that myelosuppressive effect, and you are bringing that animal to the normal blood count. So what we call as normalcy, or to reach your equilibrium. So that is important in integrative medicine. We need to study whether it has some impact of your allopathic medicines. If there is no impact, whether there is some impact with respect to other pharmacodynamic parameters, whether those medicines may minimize your side effect. So that is very good. So this study clearly shows there is no interaction of your cyclophosphamide when it is taken with the Guruji. That means when the patient takes cyclophosphamide during chemotherapy, the cyclophosphamide plasma concentration will remain same, which is responsible for the anti-tumor activity. That may not get compromised because of taking of Guruji along with it. But additionally, when you take a Guruji, it is minimizing the side effect because of the chemotherapy. And that is through, you can see that there is a restoration of your blood component that is quite clear. So that is the advantage of taking some other medicines along with the other. And that is why you can design integrative medicines in a way so that you can rationalize your treatment to prevent such kind of diseases, especially your chronic diseases. And that is why integrative medicines are coming up. And that is why I mentioned there is a lot of scope to go and study those integrative medicines at various university levels. So again, this is one study which we have done. I have done maybe 10 years back when I was doing my PhD. But there are similar studies available in the literature with respect to this kind of interaction. So interaction with respect to the pharmacokinetic interaction, pharmacodynamic interaction. So when you take a garlic with warfarin, the anticoagulant drug, so there is increasing clotting time. And there is a recommendation that you should not use garlic during your operative procedures. You should stop using garlic about 4 to 6 weeks prior to your operation. So because it has an impact on clotting time. Because we normally have to take one. So then again, Piper distinct from with levodopa is one dopamine agonist drug. It shows the antagonistic effect when you take these two medicines, these two drugs, one herbal and one allopathic medicine. And there is a recommendation you should avoid this combination in the Parkinson's treatment. So again, when you take ginkgo, one of the herbal medicines with the haloperidol, it increases the efficacy but it decreases the adverse effect associated with the haloperidol. And therefore it is showing synergistic effect and it shows the beneficial interaction. And as I mentioned, there is possibility of no interaction. So this is the case, lysozyme when taken with the midazolam, there is no impact, there is no effect, and there is no risk, no benefit while taking of these medicines. So some medicines when you show the synergistic, some medicines show the antagonistic, some medicines, some interaction shows the no impact. So these are studies there in the literature, and there are some other examples I mentioned here with respect to the pharmacokinetic interaction. So when you take one of the Western herbal medicines called as hyperacid perforatum with the cyclosporin, which is one of the anti-cancer drugs, it lowered down the blood concentration of cyclosporins, and that is why the possible mechanism is because of the enhancement of the certain CYPs and certain glycoproteins which are responsible for the influx of that cyclosporin. Another example is the pipermethrin with the propiamol and the rifampicin. This combination clearly shows that there is an increasing the blood concentration levels like Cmax, total urea under curve of these drugs, and there is decreasing your exponential time. And very famous known interaction is the grapefruit juice. When it is taken with the this pexophenidaine, there is decreasing bioavailability of these pexophenidaine drugs, and the mechanism is because of some kind of transporter proteins, there is English complex transporter protein. And this is one example where there is no pharmacokinetic interaction. So this interaction I am showcasing is, these are, it's known in the literature. So what we have done, we have studied in vitro, in vivo, some interaction which are there in the literature, which are already done in animals with respect to the pharmacokinetic interaction, with respect to the pharmacodynamic interaction. Still, some reports, those are tested in the clinical trials. So these all reports are available in the literature in a pieces, in isolated manner. So those are not in an integrated manner, those are not in one database structured way. So it is scattered information is there. So if this information is not available to the physician and to the pharmacist, so then they have to go to individual search. So search with respect to the Tinospora, then it has to go to the pharmacokinetic interaction of Tinospora, then what are drugs which are interacting with the Tinospora. So it takes a lot of time, a lot of energy. So decision is not getting, we can say, during your actual patient care, when doctor, physicians and pharmacists are there. So when there is a critical condition like cancer conditions, then you need this information readily available with you, and that is actually the power of your computational tool. So this all computational tool gather this information which is there in the existing domain in the literature, and they combine all these studies, because you can see doing one study it will take years, because one study which I mentioned, it took 1.5 years to complete it, to do in vitro studies, to do in vivo studies. So these studies we have mentioned, it also takes 1 to 2 years, because you need to do some certain clinical trial, you need to recruit the patient, you need to do the statistical power analysis to outcome of this clinical trial, and then it get published and then it available to all of us to see if this kind of interaction is there. So if we can compile all these reports in one database, that is what we call as an artificial intelligence tool is going to give us benefits. So this is what we consider the power of artificial tool. Is it gathers all the information associated with your in vitro studies, your in vivo studies, the studies from clinical trials, the studies from post-marketing studies, what we call, and then it will generate some database. It will compile this information in one database, and from this you can get some knowledge, some kind of decision makings by using this artificial intelligence tool, and this is how it helps to design and practice integrative medicines in the chronic diseases. So yes, so these studies are important, and based on these studies, there are AI tools now available to give you some decision on the organ interactions. So these are all tools is there in the literature. See the power of this tool. You can go through the reference I mentioned below. You can list all the AI tools which can use the information in one click. So for example, this is one CMSS, it is a basically Chinese AI tool, where you can see there is around 139 herbs is the information is there for the 139 herbs out of the 52 numbers of with respect to the traditional Chinese medicinal formulas, and the information with respect to 251 drugs. So you can see there are 5,400 herbal interactions reported in this database, and those are with respect to the pharmacokinetics and pharmacodynamics aspect of these herbs and drugs. So again, so you can see the number of tools available. Now this what is the mentioned in this table, these are available free of charge. You can just Google it, put the name of that database. You will get the information with respect to all these herbs, all these formulas, and all the allopathic drugs. And it is a free database. This is not it will not take some charges for this. And again, there are few examples which need some charges. But you can see there are multiple herbs listed, multiple drugs which are causing the pharmacokinetic and pharmacodynamic interaction. So this database is available. So by clicking or by just typing a name of your medicines, for example, cyclophosphamide, for example, any anti-diabetic drug, you will see whether this drug has some kind of interaction potential with some of the herbal medicines. So this is what we can see the power of the artificial intelligence. By one click, you will get all the information. So you can make your decision very fast, very quick, and you can design a medicines for those patients, especially for the chronic conditions like diabetes and cancer. So just to conclude it, integrative medicines is an evolving healthcare practice, which is we know that is basically based on the holistic approach. Herbal medicine, along with the modern medicines, are important component of integrative medicines. As we see in the in vitro, in vivo studies, especially the CYP-based studies, those can predict your possible pharmacokinetic interaction. And as we mentioned, the in vitro, in vivo, and clinical studies, which provides the basis for development of database like artificial intelligence in the integrative medicines. And of course, these tools are helping us in the understanding of possible outcome of such interaction and to take some decision on rational design of integrative medicines. So I think this is all about, yes, I mentioned that I'm not an expert or listening to talk on AI, but these studies, what we can do, the studies are important. So based on these studies, until unless you generate data, you won't able to design some AI tools. So you need to have prior art studies to make some decision, to make some develop and design some AI tools, and definitely those tools will helpful for integrative medicine for the patients which are on chronic conditions. So you have some questions.
What troubles, what obstacles did you find in your all work? Is there any challenging work you find that can you explain to all of us?
Yeah, definitely. Because this remembers my actually PhD days. So doing PhD in India, it's like you can do your technical work around 40%, but you need to do 60% of non-technical work. So you need to manage several kind of systems, I would say. But definitely we face a lot of challenges. The challenges first with respect to the establishment of assay. Assays are important because that is your input system. So based on that you are going to make some interpretation. So when we started establishment of our in vitro assays, so initially we started using a rat, and we are isolating livers from a rat, and that is what we call as a liver isotype system, so liver microsome system. So it took a lot of challenges because you need to hold the rats, you need to have some expertise to handle the animals, you need to have some expertise to isolate the organs from the animals. You need to prepare the right kind of microsome system, and you need to develop some analytical methods, because it's not easy to develop an HPLC or other kind of analytical tools, because in herbals you can say you can find there are thousands kind of different compounds. It's not a single compound, and those compounds can have some kind of interaction, some kind of interferences in your HPLC, in your any analytical tools. You need to rule out that. So those challenges is also there when you do such kind of studies. Again, when we see animal studies where we need to inject the cyclophosphamide, and that dose is very, you need to inject very small volume. You cannot inject 0.5 ml to the animal. You need to inject it not 5 ml, 50 microliter. So those small volumes, you need some expertise. You need to train on that. So that is some technical aspect. But there are certain challenges, because when we started this study, that time we don't have much studies in the literature, and there is some lacunas, I would say, in the literature, because we have correlation of in vitro to in vivo studies with respect to the allopathic medicine, because it is based on one medicine. But those rules cannot be applied to herb and allopathic medicine scenarios, because it has a different components in that. So those challenges were also there. But it remembers when we start in front of students like the PG students, we remember our days when we do our PhD work. Thank you very much.
Most of the databases are available. So what should be the criteria of selection of herb and drug? That is the first question. And second question, most of the AI tools are available nowadays. What should be the authenticity of these tools?
So just to answer the first question, what should be the criteria for herb-drug interaction? So when as a researcher, you can select any herbs and any drugs, but to design a database, you need to have some criteria, and that criteria would be, so we need to select those herbs which are routinely used in our India. So we need to list it commonly used herbs. So like an example, there are certain Ayurvedic herbs like ashwagandha. We normally take ashwagandha as an immunomodulator, and without concern to our physicians. So we need to find out, we need to list those commonly used herbs first. It may be 10 herbs, it may be 20 herbs. So that will be our list first. And also we need to see what are the commonly used allopathic medicines. So we take normally some over-the-counter medicines. We take some painkillers, commonly take painkillers, or some antibiotics which have been used for the common infectious conditions. So those we need to list. And based on those commonly used herbs and commonly used drugs, we need to plan some studies to do some herb-drug interaction. That is first. And with respect to the available artificial intelligence tools, are they valid? Are they going to give the correct or valid information? That is a question. But as per as the literature and the current knowledge of this AI with respect to the herb-drug interaction, it is evolving. It is in a transition. Tools are getting some available information in the literature. They are doing some databases, but it is not yet validated. So authenticity is a question as of now, but we can see maybe next 2-3 years we would see some authenticated AI tools for processing this. conference. I think you must have been enjoying your different research topic on AI. So this morning, and thank you very much to all participants. Maybe some academic participation, some students from some of the different colleges. Thank you for your kind patience. Thank you for listening. Thank you.
Sir focused upon was herb-drug interactions and utility of artificial intelligence in preparation of the database for the herb-drug interactions. Now, as far as the topic is concerned, as we have witnessed that in the post-COVID era, there has been a great increase in the use of several immunomodulators, dietary supplements, nutraceuticals, and much more for enhancing the intelligence will be really helpful to have an insight about this integrative medicines and herb-drug interactions. So on behalf of all the participants, delegates and organizers, once again, I am thankful to Dr. Patil sir for a very nice session. Thank you.
Thank you so much, sir. Now I request Dr. Anil sir to kindly hand over the certificate and memento. Now I request co-chairperson of this session, Dr. P.S. Patil sir. Thank you so much, sir. I am very much thankful to today's speaker, Dr.